Okay, today I'm doing a video on the somatic and visceral general senses. If you recall, uh, general senses were divvied up into the somatic senses, the visceral senses, and special senses. So we're saving special senses for later. I want to do a really sort of general, broad view of the general senses, and they detect quite a variety of senses, but they fall into categories that are detected by three of the main receptor types, and those would be mechanoreceptors, thermoreceptors, and nociceptors. Let's write those out. So again, the general senses utilize these receptor cell types to receive information of the general sense in the general sense category. And those were mechano, thermo, and nociceptors. And remember, nociceptors aren't really their own category. They're actually, there are subtypes of nociceptors that are both thermo and mechano, and as well as chemo. Oh, sorry, I forgot chemo. And chemo is going to get thrown in here as well. Chemoreceptors. And so they're going to utilize a wide variety of receptor types. And so as opposed to the special senses, which are all found on the head and are going to have very specialized receptor cells, another key point here. Remember, special senses were a two-cell, again, the cells that convey information into the CNS, right? Not the whole total number of cells involved, but the cells that convey information into the CNS. In the general senses, it's a single cell, and in special senses, it's two cells because there's a specialized receptor cell, which is a modified neuron, and sometimes it's a modified epithelial cell type that conveys information to the sensory neuron, whereas the other receptor types, the sensory neuron is the receptor. In other words, the mechanoreceptor here or the thermoreceptor here, those are sensory neurons, right? What they're receptive to is based upon filtering mechanisms or types of gated channels that they possess, right? Temperature gated channels versus chemically gated channels versus mechanically gated channels. And remember, this kind of could fit into all of those. The nociceptors can fit into any one of those three categories, really. Okay, so where do we find general sense receptors? So we'll sort of go through the gamut of locations here. I'm going to start with skin. Right? And so when we think of skin in terms of collecting information, we typically think of tactile, right? However, or touch, right? Tactile, touch, same thing. And that's true. And all of those receptors are going to be mechanoreceptors. They're going to sense some sort of membrane distortion. In other words, the free nerve endings, i.e. the dendrites of the sensory neuron, are going to have mechanically gated channels that are opened when those nerve endings are bent, essentially. Now, what bends them is based upon where they're located and filtering apparatus structures that sometimes surround them. I'll go back to another one. We talked about free nerve endings that are unencapsulated by anything, and encapsulated nerve endings that sometimes have this sort of accessory structure, usually connective tissue, sort of wrapped around the end of it. And that's just basically a filtering device that sort of limits the amount of information and type of information that those nerve endings can receive. So in touch, we have everything from pressure to vibration to, and it's like deep pressure and superficial pressure, to shearing, sort of pulling forces, and where they're located. And here I don't want to get too specific about where each type is found. There's a whole variety of encapsulated and unencapsulated nerve endings that collect this information. I don't want to get too carried about about what their names are or where they're located specifically in the skin. I don't think we need to go to that level of detail. But I do want to make sure that we understand that mechanically gated channels in those free nerve endings or encapsulated nerve endings are going to help convey the sense of touch. Now, that information is not only conveyed from each location on your skin surface, but if you think about it, our perception of it is we need to know where these things are happening on your skin surface. And so the spatial location of tactile information, tactile stimuli, has to be retained as it's sent to the central nervous system so that we can sort of map it out and know where these things are. And what happens is basically, so if I draw a really simple sketch over here, and I'll show tactile input from a sensory neuron coming on one side here. So here's some free nerve endings coming into the spinal cord. There's our cell body, right? It comes into the spinal cord. Synapses, sorry, that's a little small, but I'm trying to squeeze a lot into our page here. Going to come in and synapse on a secondary cell, and that cell is actually going to, the axon of that cell is going to cross over to the other side, right? So it's going to synapse on a second cell. Hopefully you can see that blue. It's going to cross over, and it's going to run up the spinal cord on the opposite side from the side it came in on, and it's actually going to come up into the thalamus. Just put a T on that. It's actually kind of a lobe structure, right? That's the thalamus. It's going to synapse at the thalamus on another neuron, and then that last neuron is going to carry that information to the sensory cortex. I say last, meaning last before reaching the cortex of the brain. And it's going to synapse in the somatic sensory cortex region, right? You probably learned that region in lab on the surface of the cortex. And the surface of your body is actually mapped out there in what's called a sensory homunculus, right? So this is the sensory cortex. And again, it would be this whole sort of band, right, in that region of the cortex that we're synapsing on. And so that's the region is the sensory somatic sensory cortex. The fact that it's mapped out so that input from each of the different regions of your body is sort of like kept spatially organized so that we can tell where things are coming in, where we're feeling things on our skin, means that it's organized in what's called a sensory homunculus. And meaning it looks like we could draw a picture of your body on the surface of that sensory cortex as it's mapped out to region by region. It's not anatomically correct because, as I've said before, you get a lot more sensory input from certain areas of your body than others, like your hands and face, especially your lips, have an enormous density of tactile receptors, meaning there has to be more sensory cortex, more of the homunculus associated or corresponding to that input, right? Processing for that greater amount of input. The middle of your back on that map, that homunculus, is going to appear very small, though your thighs are going to appear small because we don't need very fine resolution of sensory input from those regions. Okay, so we get this this tactile mechanoreceptor in, and again, I'm going to come back to, but the tactile would be mechano. It comes in this process. What I was getting to with the anatomy here is that it crosses over. That's called decussation. And why it does that, there are multiple theories as to why there is decussation, so we won't get into that. But it does. And so what it means is that as if this is the front of this person's, we're looking at their face essentially in front of their brain here. This would be information would be coming in from the left side of their body, say their left hand, coming into their spinal cord, going up through the spinal cord. Now, and where this decussation occurs kind of varies depending upon the type of input. But in this scenario, it's decussating in the spinal cord and then traveling up to the thalamus and then off to the sensory cortex. Well, what that overall means is that the right side of your brain receives sensory information from the left side of your body, largely, right? In other words, we get a sort of a contralateral side of the CNS is processing the information from relative to where it came in. Now, that's not true for all sensory information. There is some that is processed ipsilaterally, right? Remember, that means same side. But there's a lot that decussates, and the motor cortex here, in the right hemisphere of the brain would actually be associated with the left side of the body. Okay, so we have tactile, we have this big variety of receptor types. Again, for our purposes, we're not going to go into the specific naming and where each one is found and what it filters, but know that the reason there's a variety is so that we can separate the perception of touch from stretch, from vibration, from texture, right? In addition, side by side with those receptors, we have thermoreceptors in the skin, and there's actually both hot and cold receptors for hot and receptors for cold, helping us detect, the temperature of the things that we come in contact with. And then there are nociceptors again in the skin, and usually those are sort of sensing extremes of mechanical actions, right? Like touching you on the arm versus punching you in the arm are two different things. Not to say that you wouldn't stimulate mechanoreceptors, but there would be nociceptic mechanoreceptors that would also receive that and process that information as pain as opposed to just touch. Okay, we don't have chemoreceptors in our skin, so we're going to come back to those. Those are under general senses overall, not found in skin. So skin would be for touch, for pain, and for temperature perception. Okay, let's move on to, one other thing I wanted to mention was keep in mind the idea of adaptation, right? That was in a previous video. Adaptation, the fact that some cells cease to respond to a continuous stimulus, right, would be complete adaptation. And so there's complete adaptation, which means the receptors are categorized as phasic. And then there's incomplete adaptation, which means they're characterized as tonic. And again, the whole point here being is, do we need constant input, constant information about a particular stimulus, or can we sort of ignore it over time, at least in between changes in that stimulus? Phasic receptors allow us to ignore the information in between changes, right? Because they shut off quickly, whereas tonic ones do not. Pain receptors, again, I've mentioned this previously. Pain receptors are tonic. You are not allowed to forget pain, right? It does not, they are very, very slow adapting, and they don't adapt much anyway, period, tonic. So that the pain is more or less continuous, so that whatever's damaged, you don't go back to using, on your limb, let's say, you don't go back to using it and then damage it more. As opposed to a lot of the tactile receptors that are very phasic, right? The example I gave previously was you put a shirt on, you feel it, you feel your shirt slide on, but then, a couple of minutes later, you're not sitting there saying, there's my shirt, I can still feel my shirt, right? And if you move, you might feel it rub against your skin a little bit, right? Those phasic receptors were stimulated for a moment, and then they stop, right? So again, it's important because our general senses, we're going to find them both in the phasic and tonic categories, depending upon need, right? What we need them to do. And again, adaptive need, not teleological need, meaning is it beneficial for survival, right? Pain, it's beneficial that we be aware of it continuously so that we can avoid hurting ourselves even more so. Okay. Let's move down to another general sense, and it involves skin a little bit, but I'm going to separate it as well, and that's called proprioception. Right? Proprioception means sense of self. These are all mechanoreceptors. And what they're doing is they're allowing us to sense and we'll get to proprioception of the head, but I want to start with the limbs first. But to sense, perception of where our limbs are in space and what they're doing. Okay, and so these are found in, and we've talked about a couple of these before, right? So let's do joints here first. Sorry, not joints. Well, let's do muscles, and then we'll do joints. Right. So in proprioception, receptors within muscles included those muscle spindles and abbreviate the GTO, the Golgi tendon organ. If you recall, again, we did these previously when we were talking about a separate topic with the nervous system. Again, because they're giving sensory information from the skeletal muscles. But the muscle spindles detect length of the muscles. And even more specifically, or more dramatically, I guess, they sense change in length. I'm going to put length, delta length. Right? Remember, delta means change in. So, okay, so this is interesting because this gets back to that sort of like, so length means sort of what we call static length. In other words, if you're sitting there, sort of in the same position, you don't stop recognizing where your limbs are in space, right? Aha, this gets back to the adaptation. So we have muscle spindles that are fast adapting, and we have muscle spindles that are slow adapting. Slow adapting ones would be firing, right? Those are the tonic ones. Those would be firing continuously, even if the length isn't changing. And so what they're doing is sort of saying, this is the length of the muscles, this is the length, right? It's constant feedback, sort of telling you what your muscle length is. And so it's telling you, and again, there's more information to come, but it's telling you a bit about where your limbs are in space, even if they're not moving, right? If we only had, now changes in length, right, are telling us only when there's a change, right? When the muscle either shortens or lengthens, and then it stops firing, so they're phasic. Right? If you only had the phasic, sure, you could tell when you went from here to here. But then if you just sort of sat here and held your arm in space, a couple minutes later, you wouldn't be able to tell where your arm is, right? It would be weird, right? You'd be like, wait, where did I put that arm, right? You have to look at it to see where it is. That doesn't happen because you have both tonic and phasic receptors, right? Continual feedback as well as on-off feedback when things change. The GTO, same deal with the GTO in terms of tonic and phasic, a little variety there. The Golgi tendon organ is found basically at the junction of the muscle and the tendon, right? So tendons attach the muscle in between to the bones. And so it's in line with the contractile part of the structure, the muscle cells themselves. And so they're pulled on as opposed to the spindles, which are side by side with the muscle fibers. The GTO, the Golgi tendon organ, is pulled on when the muscle contracts. And what that means is it tells you not about length, but about tension. Right? And again, I could rate tension or change in tension, right? Because we get the ones that fire continuously and then the ones that fire on off when tension changes. But again, the two together are sort of telling us, you know, how much load we have on the limb as well as what position it's in, right? Those muscle spindles. Okay, so those are the muscle receptors. With regard to proprioception of the limbs, we also have joint receptors. Now, these have specific names, and actually they're some of the same names as some of the skin receptors. And these are nerve endings or encapsulated nerve endings that are found in joint capsules and ligaments. Right, so now we're not getting information through these joint receptors. We're not getting information about the muscle. We're getting information about just like take the muscle away, like what position are the bones in essentially, right? And so the ones in the joint capsules, the very mobile joints are encapsulated with a membrane. And then there's a set that are found in the ligaments. Remember, tendons join muscles to bones. Ligaments join bones to bones. And so as the joint angle changes, right, the tension and stretch on these ligaments changes. And so as that tension and stretch and movement of these ligaments occurs, we get sensory information that it's happening, right? And so again, we're getting information about the joint, we're getting information about the muscles, all of it helping us to tell where our limbs are in space without having to look at them and see where our limbs are in space. Okay, let's see. I think we're ready for the head, right? So we get proprioception of our limbs. How do we tell where our head is in space? And that seems like an odd statement, I guess, but right? But we don't have, I mean, other than your neck, you don't have, you know, receptors in joints that would tell you whether your head is tilted to one side or another. And you certainly don't have receptors in joints that are telling you whether your head is, you know, moving side to side or moving your whole body, but your head specifically is moving faster or slower, right? Next time you get in a car, right, as a passenger, close your eyes for a minute, right? You'll hear the car start and everything like that, but you'll also feel when the car starts to move. How do you sense that, right? That is proprioceptive receptors found in your head that are telling you about changes in velocity, right? And so the head receptors are found in what's called the vestibular apparatus. And that's really broken up into two main functional regions, right? One's called the semicircular canals. And the other is called the otolith organs. Now, we are going to punt the greater portion of the detail of this to when we start talking about the inner ear with hearing. But I want to point it out now because it really is part of proprioception. It's not part of hearing, even though it's found very closely associated with the hearing structures. Okay, but so this vestibular apparatus, again, we'll deal with the structures of it later, broken up into these two main sort of functional and anatomical units. Allow us to sense two main things. The semicircular canals are these systems of tubes, like they're a system of tubes set up in 3 different planes, just like X, Y, and Z on a coordinate axis. And what they do is they allow us to sense the movement of our head in 3 planes. So a front plane, right, a side plane, right, and a rotational plane. And so what it allows us to do is sense, we could call it axial movement of our head, or however you want to phrase that. Let's call it head movement. Right? And so input from all 3 of those different planes, right? We've got a semicircular canal associated with X, Y, and Z plane. Stimuli the receptors within each one of those little tubular systems will tell us how our head is moving in space, right? Proprioception, sense of self. You close your eyes, right? You can sense that you are moving your head in one way or another, or if it's tilted to one side or the other. And again, movement, let's not do the tilted part yet, but how it's moving in space, right? The otolith organs tell you about head position, static position as opposed to movement. Head position. And acceleration. Aha. So, in other words, these are the receptors that tell you if you're, you close your eyes and tilt your head to one side. You don't need to look in a mirror to like keep your head straight, right? If you tilt your head to the one side or front or whichever direction, you know that it's happening. And so we have right? All mechanoreceptors that are being affected by gravity, right? Head position, right? So when we tilt our head, we're going to stimulate these receptor cells, mechanically stimulate them due to forces of gravity, which we'll show you the filters that allow that to happen later, but that are going to cause ion currents, receptor potential in those receptor cells, neurotransmitter release, and off we go into the CNS, right, telling us about our head position. Now the acceleration is a different set. Those are the otoliths. The head position is in the otoliths, and the acceleration is in the otoliths, right? Head movement. But the acceleration bit allows us to sense both linear acceleration, the example I gave you about being in a car, or vertical. So you actually have 2 sets, right? One that gets stimulated with you like go up or down in an elevator, right? You, again, get on an elevator and close your eyes, you know when it starts moving. And so you've got 2 sets there to sort of sense your changes in velocity, right? To tell you about your body's movement in space. More so, acceleration is changes in movement, right? Changes in speed. And so we've got proprioception from the muscles and the joints, which collectively we would say limbs, and then proprioception, right, relative to the head. Okay. Lastly, I want to bring in the visceral senses, right? Because we did the touch and pain and temperature of the skin. We've got proprioception from the limbs and from the head. And I've run out of room, so I'm going to jump over here. But we have to talk about the visceral senses a little bit. Okay. And the visceral sense, now pain is one of them, right? That's a possibility. There are thermoreceptors that are part of the visceral senses. We have thermoreceptors not associated with skins. They're found in your core. You actually have some in your brain as well. We have, what am I missing? Mechanoreceptors. Now, those guys, I'll clarify on those a little bit. Mechano is a very general term. With regard to visceral function, we're going to put them into a we're going to define them a little bit more closely. But in addition to the mechano, we have chemo. Right? So a whole lot of input from the visceral senses. Now, where do we find, so I'm going to focus on these. These guys are pretty straightforward, right? They're sensing temperature of the core, they're sensing pain of the visceral organs. The mechanoreceptors are called baro or stretch receptors. And what this means, and if you think about baro, what's a barometer measure? A barometer measures barometric pressure. And so these are pressure receptors. And we have another version that I'll call stretch because it's not so much pressure because the pressure is very low. And these guys are found in some very specific regions, and they're largely about, well, there's a cardiovascular set that's largely about telling you about blood pressures, right? And so they are found in the aorta. That's the largest artery carrying blood right out of the heart to your whole body, minus the lungs. They are found in the carotid sinus. So the carotid arteries travel up your neck, and then they branch to go inside to your brain and then outside to your scalp. And right at that branching point between an internal and external, there's this region called the sinus. It's just sort of an expanded area where the carotid artery branches. And so we've got receptors there. And we've got, so these cardiovascular stretch receptors are found in the large veins, atria / ventricles of the heart. So the chambers of the heart, really, you could say. And so you might say, okay, well, what are these for? Well, let me back up. So the aorta and the carotid sinus, we have baroreceptors there because we need to make sure that we know that we have sufficient blood pressure to drive blood flow to our brain. Carotid sinus ones are, right, going to the head. And then in general, in the aorta, right, this is sort of like backup. The carotid sinus is like, okay, your brain's really important. Make sure that we have receptors there. The aorta is just sort of like, that's going to everything. And so we have receptors there telling us about blood pressure driving flow to all organs. And because pressure drives flow and we need flow to keep the tissues alive, i.e. enough oxygen delivery, things like that, we're going to have receptor input there that allows us to know our blood pressure and then therefore regulate our blood pressure so that we can maintain our blood flow. The ones that are found in the large veins and the atria and the ventricles, well, this is on the other side of the circuit, right? This is in the blood coming back to the heart, right? And literally into the heart. Well, and the reason I put this term stretch is because the pressure in those large veins and the heart, as it fills, not when it's contracting, but in between contractions, is almost zero. So you can't really measure the pressure. They're not pressure receptors so much as stretch receptors, like distension receptors. And what they'll allow us to do is say, is our heart filling appropriately, right? Do we have enough blood volume coming back to the heart to distend it appropriately, fill it appropriately? And again, that'll be linked to receptors that might help us, regulate our blood volume, right? Make you thirsty, right? If you don't have enough filling, and among other things. But we would adjust fluid intake and fluid output so that our blood volume is adjusted to maintain the volumes in the large veins and the heart as it fills to ensure proper function, right? And so again, they're mechanoreceptors that are sensing pressure or just distension, filling, if you will, within the cardiovascular circuit. Now, if we go to the visceral senses, you could also think of mechanoreceptors as being found in like your bladder, right? How do you know when your bladder is full? Well, stretch receptors in the wall of the bladder. How do you know when you just ate a meal and you're full, right? Stretch receptors in the wall of the stomach. And there are stretch receptors in the wall of the lung that tell you how well your lungs are inflated, like literally during a breath. Right? Are they filling up? Should we start to exhale soon? Right? Stretch receptors are giving you that information. Again, unconsciously giving you that information. Okay. baroreceptors. Good. Now we need to do the chemoreceptors and their locations, and they're pretty straightforward because they're almost the same. Because they're also the aorta, also the abbreviate carotid sinus, but now the third location is not in the large veins; it's in the medulla of the brain. All right, well, what are these chemoreceptors sensing, and what are we using this information for? Well, and there are other chemoreceptors, but the ones in the visceral senses that I'm referring to here sense O2, CO2, and literally H+, which I'm going to put down here as pH, because the measure of the amount of hydrogen ions, H+, that measure is called pH, right? And so these chemoreceptors are sensitive to O2, CO2, and pH. They might be, and again, there's different locations for these, and in these different locations, they might be sensitive to one or the other of these more so. There is a difference in what they're receptive to. And what they're allowing us to do is sense our blood or cerebral spinal fluid, right, of the brain, and the brain sort of interstitium, the interstitial space of the brain, to say, okay, do we have enough oxygen? Are we moving enough oxygen through our body, getting enough from our lungs, and moving it through the cardiovascular system to maintain homeostasis, right? If CO2, if we're not exhaling enough CO2, we're not moving it out of the body fast enough, this will accumulate. If this accumulates, it hydrates and actually releases protons. Aha, so pH would actually go down, right, as hydrogen ions accumulate. And so if you think about it, right, this is kind of like a metabolism indicator, right? It's sort of saying, right, if, for instance, you start to exercise, oxygen consumption goes up, so this goes down. CO2 production goes up, so this would rise. Because of this rising, this would go down. Those three changes are sensed by chemoreceptors to adjust, and that input is used to adjust, guess what? Cardiac output, how much blood your heart is moving, or the speed with which you ventilate your lungs, how rapidly you're breathing, all in an attempt to restore homeostasis, right? And so we have these guys sensing the blood and sensing the brain, chemical conditions, to make sure that our pulmonary breathing and cardiovascular moving those blood gases around is sufficient to maintain homeostasis, right? And so our chemoreceptive sense for visceral function is all about homeostasis. We'll talk about chemoreceptors when we get to special senses, right? You have olfactory, sense of smell, tongue for gustation, taste. Those are chemoreceptors too, but they're not about homeostasis, right? These are about maintaining homeostasis internally as opposed to sensing external things in the air or the food you're eating. Okay, that's the general senses with proprioception and visceral senses and the whole gamut.